Welcome to the CIBM Center for Biomedical Imaging Seminar Series number 6. It is my great pleasure to introduce you to Hélène Lajous, who is a research staff scientist at the CIBM Signal Processing CHUV-UNIL section, who are focusing on computational neuroanatomy and fetal imaging. Hélène will be sharing with us T2 quantification from super-resolution reconstructed clinical fast-spin echo MR acquisitions. Thank you, Hélène. The floor is yours.
Thank you, Pina. Good morning, everybody. Thank you for being there, and thanks, Pina, for this nice opportunity to present part of the work we conduct at the Medical Image Analysis Laboratory. So I'm Hélène. I'm an engineer in biomedical imaging and a PhD in biomedical technologies. I joined the CHUV-UNIL signal processing section of the CIBM last year as a postdoc to work on fetal brain quantitative MRI. Today, I will present you our strategy for super-resolution T2 mapping from fast magnetic resonance acquisitions. As you may know, MRI is a powerful tool complementary to the ultrasound gold standard in the follow-up of the fetus, as its high resolution enables accurate biometric measurements. MRI is especially used to investigate equivocal neurological patterns. In clinical routine, fast-spin echo sequences are used to avoid unpredictable motion during acquisition. In practice, at least 3 orthogonal series of 2D6 slices are acquired during breath hold to ensure a good signal-to-noise ratio and to provide information on the whole volume of the fetal brain in around 30 seconds. For the past few years, MIAL has developed advanced super-resolution reconstruction techniques for fetal brain MRI. So several series are required in orthogonal orientations. At this point, they may still be corrected by motion. Then the low-resolution series are aligned to a high-resolution volume. Since there is redundancy between the different series, it becomes possible to reconstruct one single high-resolution volume with an isotropic resolution of 1 millimeter. So from the original 3 millimeter thick slices, we obtain a super-resolved image of the fetal brain. The super-resolution algorithm developed by Sébastien Tourbier and colleagues relies on the classical formulation of an inverse problem with a data fidelity term. Since it is ill-posed, we used a spatial penalty, here total variation. Super-resolution reconstruction of the fetal brain makes it possible to get more accurate information on the structure of the developing brain. But we now want to go further into the details of the processes at work during brain maturation. Fast changes have been observed during brain development. Quantitative measurements have been mainly reported in preterm and late-term newborns, showing an increase in fractional anisotropy and a decrease in T1 and T2 relaxation times after birth. However, there is no ground truth on fetal brain measurements yet. So the combination of multiple multimodal biomarkers specific to the ongoing maturation processes would provide doctors with new tools for evaluating pregnancies as a support for diagnosis and prognosis, but also as for postnatal management. This would also allow to link prenatal and postnatal evaluation. Besides, such an insight into in utero early brain maturation would make it possible to identify new microstructural biomarkers associated with abnormal developmental patterns. So in this context, we aim at developing a new method for 3D super-resolution T2 mapping of the fetal brain. In the following, I will present you a new scheme for quantitative MRI of the developing fetal brain. The results of this in vitro feasibility study have just been accepted for the International Conference on Medical Image Computing and Computer-Assisted Intervention. Then I will give you an overview of the numerical phantom of the fetal brain we are currently developing for validation and further investigation. So let's start with the in vitro work I conducted to demonstrate the feasibility of our T2 mapping strategy. Spin echo images are usually acquired at various echo times to measure the decay of the transverse magnetization over time and deduce the corresponding T2 relaxation time of the tissue. So from this technique, we decided to acquire the fast spin echo haze sequence used in clinical routine for fetal brain examination at different echo time. A super resolution volume is then reconstructed from various orthogonal series by solving the inverse problem described earlier for every echo time. And finally, a T2 model fitting is run on the resulting images. To demonstrate the feasibility of our method, I acquired data on a quantitative MR phantom, the NIST phantom, that was originally developed to assess the reproducibility and repeatability of MRI protocols across vendors and sites. The NIST phantom is made of several layers and resolution insets. I focused on two layers that consist of elements of various T1 and T2 properties. So some of them have T2 values in the range we expect, so between around 170 milliseconds and 450 milliseconds. T1 values are substantially lower than the 3 seconds that were reported in the white matter of the developing fetal brain. So for consistency, I decided to focus on only three regions of interest that exhibit T2 values consistent with fetal brain properties and high T1 over T2 ratio, and that can fit in a similar field of view as for fetal brain examination. I evaluated our 3D super resolution framework with respect to T2 maps obtained from low resolution haze acquisition and from reference sequences where only one thick slice was imaged. So repeated single echo spin echo sequences at various echo times and multi-echo spin echo sequence. As mentioned earlier, I focused on a restricted field of view containing only our 3 regions of interest. The acquisition time of the reference single echo spin echo sequence at variable echo time exceeds 7 hours. It was drastically reduced to around 16 minutes in the case of the MESE. However, such a long acquisition time remains completely unreasonable in a fetal context. Each haze series required about 28 seconds of acquisition. So I first computed voxel-wise T2 maps by running a T2 fitting within every voxel of the image. Then I performed a region-wise analysis on our 3 regions of interest to deduce mean T2 values over every row. And finally, I computed the relative error in T2 estimation using single echo spin echo measurements as a reference. So here you can see examples of voxel-wise T2 maps obtained from reference sequence. So first the single echo spin echo using 25 echo times, the MESE using 32 echo times, and then the haze, so here in a configuration with 18 echo times and the corresponding super resolution reconstruction. So even though I show you the result for only one coronal slice here, recall that our technique makes it possible to compute 3D T2 maps in a clinically reasonable acquisition time. I ran the same MRI protocol on 3 different days to study the repeatability of T2 measurements from every method and to assess the error caused by variations, for instance, variations in temperature, in phantom positioning, or scanner calibration. Here are the results for the different configurations I tested. So I know there are many numbers and we do not have time to go through all of them now, but I would like to draw your attention to specific points. So first, bold value shows the minimal error in repeatability for each method and in every row. On average, our method is highly repeatable over independent measurements with a relative difference in T2 estimation, less than 5% for the haze and less than 10% for the super resolution reconstruction. If we now put things back in the context of fetal imaging, acquiring at least 3 orthogonal series using 18 different echotypes doesn't seem feasible in clinical practice. Thus, if we focus on a more realistic intermediate configuration, for instance, using 6 echotypes, we can see that the coefficient of variation is less than or equal to 1.5% for haste-based quantification and less than 4% for the corresponding super-resolution strategy. According to the literature, these discrepancies seem acceptable. So overall, our strategy enables repeatable T2 quantification in reasonable acquisition times. In an effort to optimize the acquisition scheme, especially regarding reasonable acquisition time, but also cell deposition in the fetus, I investigated the influence of the number of echo times on the T2 estimation accuracy. Since T2 measurements are highly repeatable throughout independent measurements, I arbitrarily selected one experiment to perform this study. Bland-Altman plots demonstrate the agreement between haste or super-resolution based T2 quantification and single echo spin echo reference measurements in the 3 regions of interest. So as you can see in green, average haste-based T2 quantifications errors are almost the same across all configurations independently of the studied ROI. Conversely, in orange, super-resolution-based T2 quantification errors vary depending on the targeted T2 value. Overall, you can see that super-resolution-based T2 mapping exhibits the smallest average difference for 6 echotypes. If we now look at the corresponding box plots that displays a relative error in T2 quantification, so first you can see on the right that the MESE provides T2 estimation with a small dispersion that reflects a mean relative error of about 16% compared to single echo spin echo measurements. However, recall that the out-of-plane resolution is low and scanning times are prohibitive. Again, in green, you can see that haste-based T2 quantification overestimates T2 values in our range of interest by around 25%. And in orange, you can see that overall super-resolution-based T2 quantification substantially outperforms haste with a mean relative error less than 14% that vary with the number of echo times acquired. So for less than 6 echoes, the super-resolution shows a dramatic improvement in T2 quantification over the haste-based technique, but only for regions of interest that exhibited T2 values less than 200 milliseconds. Above 6 echoes, both super-resolution and MESE have similar error in range. And finally, for the configuration using 6 echo times, super-resolution substantially outperforms both haste and MESE over the whole range of T2 values studied with a relative error less than 11%. Finally, I evaluated the robustness of our method in the presence of noise. So in clinical scans, I measured an average signal-to-noise ratio in the range of 300 to 400, which was found similar in phantom acquisition with an effective echo time of 90 milliseconds. But now the question is, how would noise affect the robustness of our method? So to study this, I denoised the original haste images using a non-local means algorithm. Then I added several levels of ratio noise to the images before super resolution reconstruction and T2 finting. So here you can see an example with a signal to noise ratio decreased by a factor of 7. Here are some preliminary results on data acquired at 6 echo times that demonstrates that our T2 mapping technique is robust, even with a very low signal to noise ratio. So I demonstrated that our super resolution T2 mapping technique provides repeatable, accurate, and robust T2 quantification in vitro. It relies on fast acquisitions that are routinely used in fetal brain examination and enables 3D high-resolution T2 mapping. So now, what's next? Our method relies on two common simplifications, a single component tissue model and a mono-exponential decay. Obviously, brain tissue has multiple components within a voxel, hence a multi-exponential model would be more appropriate. However, fitting multiple compartments would lead to a nilpose problem that requires high signal-to-noise ratio and acquisitions with many echo, which means, again, unreasonable acquisition times in a fetal context. Another limitation is linked to motion. So as it was not possible to expose our phantom to control translational or rotational motion, our study is limited by the lack of data in the presence of movement, whereas unpredictable fetal motion is a major hurdle in fetal scans. So therefore, simulations give a framework for further investigation and validation. And which leads me to give you a brief overview of the numerical fetal brain phantoms that we are currently developing in collaboration with Christopher Roy from Matthias Stubel's group and Tom Hilbert from Siemens. So here the idea is to simulate fetal haste acquisitions to further explore the potential of our 3D super-resolution T2 mapping strategy, especially in the presence of motion. So first we need to understand how MR acquisition of the fetal brain work. Schematically, the mum-to-be is positioned in the scanner. The radiology technician defines a large field of view that covers the entire fetal brain. In the presence of the main magnetic field, protons of water molecules align themselves in the direction of B0. Several RF pulses of different amplitudes are then successively applied to disrupt the balance of the proton and to measure their return to equilibrium, namely the relaxation. The T2 relaxation time is defined as the time required for the transverse magnetization to fall to approximately 37% of its initial value. Obviously, this time depends on the nature of the tissue. So then two additional gradients are applied in order to spatially encode where the signal of the protons come from. Frequency components are acquired in what we call K-space, basically the Fourier domain. And finally, the image is recovered by inverse Fourier transform. So as a starting point for our simulator, we need a fetal brain model. I chose to use segmented high-resolution anatomical images of the fetal brain from Golipul's normative spatiotemporal MRI atlas. So this atlas gathers reference high-resolution images of fetuses from 21 to 38 weeks of gestational age. Since we lack ground truth measurements in the fetal brain, as I already mentioned, I decided with the help of our collaborators, Dr. Vincent Dunet and Dr. Miriam Cobb, to classify all the segmented tissues into only 3 categories, so gray matter, white matter, and cerebrospinal fluid. Then I assigned T1 and T2 values to the corresponding anatomical structures according to the literature in order to get reference T1 and T2 maps. I then used the extended phase graph concept, a powerful tool for depicting the magnetization response of a broad variety of ML sequences that especially provides in-depth description of T2 relaxation processes. So from the pulse design of the haste sequence, the T1 and T2 reference maps, and a simulated B1 bias feed, I can compute the true decay of the transverse magnetization within every voxel of the images, leading to a 4-dimensional matrix. So that combines both the temporal information and the anatomical information. We are now ready to address K-space sampling of the images to be simulated. So as I said, we computed the T2 decay in every voxel of the anatomical images. By applying a Fourier transform to this 4D matrix, we obtain as many frequency domains as there are sampling time points. And then at most one line in every of these frequency domains will be used to fill the actual cast space of the simulated haste images. So first, the center of cast space is sampled with a central line of the frequency domains that corresponds to the effective echo time of the haste sequence. Then reference lines around the center of cast space are sampled. They correspond to low frequency components. For further lines, an acceleration factor of 2 is simulated by using only every other line to fill cast space. And since haste acquisition is a partial Fourier technique, slightly more than half of cast space is actually sampled, as schemata is here in blue. So the complementary part is artificially fed using Hermitian symmetry. So if I summarize, from segmented high-resolution anatomical images of the fetus, of the fetal brain, we perform tissue classification between gray and white matter and cerebrospinal fluid. Then we convert this anatomical information to MR contrast in order to obtain T1 and T2 reference maps. Then, using the extended phase graph simulations, we can compute the T2 decay within every voxel of the anatomical images, which helps us in sampling cast space. Then, random complex Gaussian noise is added to cast space to simulate thermal noise during acquisition. And finally, haste images are recovered by inverse Fourier transform. And here's how the simulated images look like. So here you have the reference high resolution images from Golipo's atlas. Here you can see the low resolution simulated haste images in 3 orthogonal orientations. And here the corresponding super resolution reconstruction from these 3 series. However, I have to say that in clinical practice, it often happens that more than 3 series are required with a slight shift of the field of view in each orientation in order to collect as much information as possible. So I simulated this shift in each orientation in order to obtain 6 series and compute the corresponding super resolution reconstruction. And as you can see, the resulting image is much sharper than when using less series. So we now have a tool that offers many possibilities, generation of images at various echo times for different fetal images, etc. So if I come back now to the first part of my talk, here is an example of how we can use it as a validation framework for our super resolution T2 mapping strategy. At the left, again, you can see the reference T2 map based on the high resolution images from Golipo Atlas. Then you can see a T2 map obtained from the simulated haste images in the axial orientation. Then the corresponding super resolution from 3 series and from 6 series based on the simulated haste images. So since the T2 value in the cerebrospinal fluid is very high, let's slightly adjust the scale in order to have a closer look at what happens in white matter and gray matter. Again, as you can see, the super resolution reconstruction from 6 series is sharper and looks closer to the ground truths. So these are preliminary results, but they show that this new tool provides a whole playground for further investigation and is a valuable framework for validation. So in conclusion, I've demonstrated the in vitro feasibility of our 3D T2 mapping strategy from fast acquisition, that are routinely used in fetal brain examination. This relaxometry technique enables repeatable, accurate, and robust T2 quantification in the range of T2 values reported in the developing fetal brain. I also described you the simulation framework we are currently developing for further investigation and validation of our super resolution T2 mapping strategy. And what about the perspectives of this work? So obviously, I want to add motion to simulate real fetal acquisition and evaluate the robustness of our T2 mapping strategy in the presence of motion. I would also like to conduct a pilot study on an adult brain to demonstrate the in vivo feasibility of our technique. And instead of a sequential scheme, another way to approach super resolution reconstruction for T2 mapping would be to perform simultaneous T2 fitting and super resolution reconstruction by adding to the inverse problem described earlier a temporal regularization that accounts for the transverse magnetization decay within every voxel. Finally, I plan to develop advanced T1 and T2 mapping acquisition schemes based on the free-running framework that has been developed in Matthias Stuber's group for cardiac MRI. With this, I would like to thank my great fetal team, as well as all my collaborators, especially Tom Hilbert for his constant support, and Christopher Roy for his help in the implementation of the numerical phantom. I would like also to thank the Swiss National Science Foundation for its support, and thank you all for your attention.